The causes of yield variation are mainly due to branching behavior, dimension of shoots, internodes length, etc. Whatever may be the canopy structure, small shoots less than 1.0 cm basal diameter at maturity, i.e. 0.7 cm during inoculation, are not good for lac production due to high lac mortality on such shoots. By and large, such shoots, 60–75%, are unproductive for brood lac. Interception of light can be facilitated if these unproductive shoots are removed. So, by removing such shoots, in one hand we can create good environment for lac growth on bigger shoots, and on the other hand, wastage of brood lac can be minimized. Similarly, in case of Semiolata, shoots having basal diameter of approximately 6 mm may be removed for facilitating better ventilation. 422. 423. Shoot characteristics. Lac production capacity is dependent on the physiological characteristics of host species and on nutrient supply to the host. Texture and size increases with the age of the shoots. Under normal soil fertility condition, when lac cultivation is practiced regularly on shoots with basal diameter ranging from 1.0 cm to 2.5 cm are considered to be the best for different lac hosts. In general, lac survival % living lac incrustation to length of settlement increases with increase in diameter, but length of lac insect settlement decreases as the diameter of the branch increases and tend to settle on secondary branches. Usually, an increase in the diameter of the branch between broodlac inoculation in July to harvesting in February is about 35% in case of beer. Therefore, at the time of inoculation, branches with diameter varying from 0.75 cm to 1.85 cm are considered to be optimum. Normally, best age of shoots for kusum, beer and palas are 18, 6 and 7 months. Best shoots are neither very tender nor very old. At proper age, certain portion of it assumes a characteristic grayish color. Lac insect incrustation grown on it remains attached very firmly and does not detach from stem. Good quality broodlac with smooth incrustation are produced from such shoots. 17.5 strains of lac insects. Rangeeni and Kusumi are two strains of lac insects recognized in India. Both strains complete their life cycle twice a year, but their maturity season differs greatly. In a year, there are 4 lac crops named after Hindi months. The crops are shown in the table given below. Strains of lac crop: weatherlac host plant. Seed. Inoculation: Devlapki, K crop. Harvesting time: in month. Rangini. Katkarani. Season: palas June, July, October, November 4. Basiki. Summer palas October, November. 424. Lac insects live in cavities or cells made in resin or lac secreted by their host plants. The average female lays 200 to 500 ready-to-hatch eggs per cycle. A mature embryo is inside the eggs when they are laid, i.e. it is fully developed. Lac insects reproduce ovoviviparously. Insects lay 3 types of eggs: I, from which males emerge in greater numbers than females; E, from which females emerge in greater numbers than males; and E, from which both males and females emerge in equal numbers. The tiny crimson red first instar nymphs emerge from the lac incrustation within a few hours of laying the eggs. The nymphs have 6 anal setae, 2 compound eyes, and 2 pairs of antennae. Nymphs in their first instar are known as crawlers. During swarming, nymphs emerge for 5 or more weeks at a time. Upon reaching soft, succulent twigs, the nymphs settle down, thrust their needle-like proboscis into the bark, and begin sucking the sap. The nymphs settle on twigs that are neither too soft nor too hard, and they settle at a rate of 200 to 300 insects per inch, depending on the number of broods they have. In a few days, after settling, the nymph begins secreting resin from glands under the cuticle, except near the mouthparts, breathing pores and anus. A semisolid resin is secreted, and when exposed to air, hardens into a protective coating called lac. Before reaching maturity, nymphs molt 3 times inside their cells. The duration of each instar depends on several factors, including temperature, humidity, and host plant. A nymph loses its legs, antennae, and eyes after its first molt, but the number of its anal setae increases from 6 to 10. At this stage, the male and female cells can be distinguished, the male being slipper-like with an operculum at the rear and the female being nearly globular. Male nymphs regenerate their appendages during their second molt, while females feed, grow, and secrete resin during their pupal stage. A male emerges as an adult with wings and without wings after his third and final molt. In the second generation, only wingless forms are born, and the males mature half as quickly as the females. As soon as they emerge, they start mating with as many females as possible and die due to atrophy of their mouthparts. Females remain inside their lac cells and start growing rapidly. Female cells increase in size so that they contribute the bulk of the lac. Lac cells become globular as they grow, filling the entire space. In preparation for laying eggs, they shrink in size to make room for the eggs. In the rear end of the cell, 2 yellow spots appear. As the spot grew, it became orange in color. Under the tail ends of the females, a large number of eggs are oviposited in the space called the ovesic. Eggs in the ovesic appear orange, which indicates that they will hatch within a week. For inoculation of new trees, the twigs brood lac are cut from the tree. 17.7 Lac crop management. It is a complex process to cultivate lac. In order to cultivate lac, it is necessary to take good care of the host plants, propagate them, and collect the milk. Lac cultivation involves the following steps. 1. Selection of site and the host plants. 2. Pruning. 3. Inoculation. 4. Swarming. 5. Harvesting of lac site selection and the host plants. A good host plant selection is crucial to lac cultivation, since the quality and yield of the lac are directly proportional to the number of host plants. Babool, pipal, palas, kusum, care, etc., are some of the host plants. Thus, the cultivation site should have more and more host plants. 425. Plants that host lac insects must be fast-growing, have low sap density, and able to withstand heavy infestation. The lac from Kusum Schleicheratrihuga attracts the highest market price of all the plants listed above. Then there are the host plants such as Pallas bufifrondosa, Beer, Ziziphus jujuba, and Cereus albizia. Pruning. Lac production is also influenced by the time of pruning. Pruning must be done properly so that the host plant does not lose health and nutrition, and also produces better quality and quantity of lac. Lac insect inoculation is usually performed 6 to 12 months prior to pruning. If inoculation is to be done in June-July, pruning should be done in January-February. If inoculation is to be done in October-November, pruning should be done in April-May. Pruning should take care of the following points. Pruning should not be excessive. As a result, the tree's general health and strength are maintained. Only cut the old branches of the host plant. It is prohibited to cut branches larger than 2 inches in diameter. Make sure the tree is shaped well so that new shoots can grow freely. Thin branches should be cut close to the trunk from which they arise. Branches that are dead or diseased should be removed. Split or broken branches should be cut below the split or break. Lac-bearing branches should not be cut indiscriminately. Inoculation. Inoculation is the first step in cultivating the lac insect. It is the process by which the young ones become properly associated with the host plants. There are 2 methods of vaccination: natural inoculation and artificial inoculation. Natural inoculation occurs in normal routine and is a very simple process where swarmed larvae infect and suck out sap from the same host plant. Artificial inoculation involves cutting twigs with insect larvae that are about to swarm. In order for the larvae to swarm to the new host plant, 426. The cut pieces are tied to fresh trees. As a general rule, artificial inoculation is considered to eliminate all possible drawbacks of natural inoculation. Swarming. Lac insects swarm during their swarming phase. Yellow spots appear on the upper surface of the anal region during swarming. Muscles contract at this stage, and the insect is detached from its attachment point. A hollow cavity is left behind, which is later filled with resin. Experience can be used to determine the exact time of swarming. Harvesting of lac. Harvesting is the process of collecting ready lac from a host tree. There are 2 types of harvesting. Immature harvesting. Lac harvesting before swarming is called immature harvesting. Ari lac is the lac thus obtained. Insects may be destroyed during harvesting, which leaves cultivators at a great loss. Only Pallas host plants should be harvested for Ari lac. Mature harvesting. Mature harvesting occurs after swarming. Mature lac is thus collected. Lac is most abundant when twigs are harvested while the females are still alive. It is possible to harvest twice a year. Lac and eggs are carried by a twig called a brood lac stick, and the lac itself is called brood lac or stick lac. Lac cultivation is a complex process. Lac cultivation involves taking care of the host plants, propagating the seeds, and collecting the lac. Lac cultivation involves the following steps. 1. Site selection and host plant selection. 2. Pruning. 3. Inoculation. 4. Swarming. 5. Harvesting of lac. Selection of site and the host plants. Lac cultivation relies heavily on the selection of suitable host plants since quality and yield are directly proportional to the number of host plants. Babool, Pipil, Pallas, Kusum, Care, etc. are some of the host plants. As a result, the cultivation site should have a greater number of host plants. It is necessary that the host plants grow quickly, have a low sap density, and be able to withstand heavy infestations of lac insects. In the market, the lac from the host plant Kusum 427. Schleicheratrihuga commands the highest price. There are also host plants like Pallas bufifrondosa, Beer, Ziziphus jujuba, and Cereus albizia. Pruning. Lac production is also influenced by the time of pruning. Pruning must be done with proper care so that the host plant does not lose health, nutrition, and also produces better lac quality and quantity. Lac insect inoculation is usually performed 6 to 12 months prior to pruning. If inoculation is to be done in June-July, pruning should be done in January-February. If inoculation is to be done in October-November, pruning should be done in April-May. Pruning should take care of the following points. Pruning should not be excessive. As a result, the tree's general health and strength are maintained. Only cut the old branches of the host plant. It is prohibited to cut branches larger than 2 inches in diameter. Inoculation. Inoculation is the first step in cultivating the lac insect. It is the process by which the young ones become properly associated with the host plants. There are 2 methods of vaccination: natural inoculation and artificial inoculation. During the natural inoculation process, swarmed larvae infect and suck out sap from the same host plant again. Artificial inoculation, on the other hand, involves cutting the twigs containing the insect larvae that are about to swarm. The larvae are tied to fresh trees so that they swarm to the new host plant. In general, artificial inoculation is considered to eliminate all possible drawbacks associated with natural inoculation. Swarming. Lac insects swarm during their swarming phase. Yellow spots appear on the upper surface of the anal region during swarming. Muscles contract at this stage, and the insect is detached from its 428. Attachment point. A hollow cavity is left behind, which is later filled with resin. Experience can be used to determine the exact time of swarming. Harvesting of lac. Harvesting is the process of collecting ready lac from a host tree. There are 2 types of harvesting. Immature harvesting. Lac harvesting before swarming is called immature harvesting. Ari lac is the lac thus obtained. In this process, the insects may be destroyed during harvesting, causing the cultivators great loss. Only Pallas host plants should be harvested for Ari lac. Mature harvesting. Lac is harvested after swarming is called mature harvesting. Mature lac is thus collected. The highest yield of lac is obtained by harvesting twigs while the females are still alive. It is possible to harvest twice a year. Lac-bearing twigs along with eggs are called brood lac sticks, and lac itself is called brood lac or stick lac. 17.8 Natural enemies of lac insects and their management. Lac enemies pose a challenge to lac culturists, as they not only reduce lac insect population, but also retard lac production and quality. There are two types of damage caused to lac insects, A. Damage caused by insects, B. Damage caused by animals other than insects. Lac crop enemies include predators and parasites. Chalicides are the most common parasites of lac insects. They are small, winged insects that lay their eggs inside the lac insect's coat either on the body of the insect or inside it. Upon hatching, larvae feed on lac insects, causing their deaths. About 5–10% of the lac crop is destroyed by this parasite. Predators cause more damage, 35% of the total destruction. The lac insect is primarily preyed upon by Euglem amabilis and Holococeras pulveria. Termites not only eat lac insects, but also destroy the lac they produce. Aside from insects, squirrels, monkeys, rats, bats, birds, woodpeckers, man, etc., are the enemies that destroy the lac crop. In addition to climatic factors like excess heat, cold, heavy rain, and storms, faulty cultivation methods also cause damage. 429. Control. Damage caused by the above-mentioned animals can be reduced to a certain extent by the use of the following methods. 1. Cultural method. The amount of damage by infection can be reduced to a greater extent by taking care during the culture of lac insects, especially at the time of inoculation. The brood lac showing the minimum enemy attack should be selected for inoculation and should be cut from the host plant very near to the time of emergence of larvae, about 1 week before the emergence. This will reduce the chances of parasite attack on the emerging larva at new place, host. The brood lac used for inoculation should be removed from the new host's branches as soon as the emergence of larvae stops, approximately 3 weeks after inoculation. It reduces the chance of transference of enemies to the new host plant from the brood lac. The infected brood lac not fit for inoculation or the used up brood lac should not be retained for long. The lac should be scrapped at once and the rest may be crushed or dropped into fire in order to destroy the predators and parasites. The delay M processing also gives chances to the enemy insects to escape into field. So the manufacturers should try to convert stick lac into seed lac as soon as possible. By these cultural methods the future production can be saved from infection to some extent. 2. Artificial method. During the crop reaping, it is not always possible for the manufacturers to convert the huge amount of stick lac to seed lac at a time. To avoid the spreading of enemies at this time from stocked stick lac, simple artificial method can be used. Bundles of stick lac should be tied with stones and immersed in fenced water, river or ponds, for about a week. This kills all the parasitic and predator insects as they cannot survive in water. 3. Biological method. It is an indirect method for killing the parasitic and predator insects. For this purpose hyperparasitic insects are used which attacks the parasitic insects of lac and kill them. These hyperparasitic insects are however, not harmful for lac crop. 430. 17.9 Lac extraction. Lac is processed after it is harvested from the host plants. The first step in processing stick lac is scraping it off the twig. This scraped lac must now be cleaned of impurities such as dead lac insects, eggs, and coloring matter. Finally, hand-operated mortars are used to crush the lac after it has been cleaned. Crushed lac is air-dried, resulting in pale yellow granules. Seed lac is what it is called. Lac incrustation. After soaking, seed lac is washed, dried in the sun, bleached, and heated to melt on charcoal in cloth bags. As the bag is heated, the lac is squeezed out, leaving the impurities in the bag. Curry lac is the name given to this lac. To form button lac or pure lac, curry lac is allowed to cool and solidify around button-shaped forms. Sheet lac is made from pure lac stretched into thin sheets. When dissolved in water, sheet lac produces shell lac, which is white or orange in color. Lac quality is determined by the host plant. A kusumi lac is said to be the best and most expensive lac, while a dock lac is said to be the worst and cheapest. Lac quality and color vary depending on the presence of gum and resin in the host plant. Types of lac. The immature lac harvested from the host plant is known as ari lac. A stick of mature lac is harvested from a host plant in the form of a stick lac. The seed lac is obtained by removing the lac from the stick and washing it. After grinding seed lac, dust lac is obtained. Shellac is prepared by heating seed lac and dust lac together. 431. 17.10 Summary. Lac culture is a lesser known, but economically sustainable, part of animal husbandry. During its life cycle, the lac insect, a small insect of the order Hemiptera, secretes resinous material. Lac is produced by larvae and pupae. The only commercial resin derived from animals is lac. A lac culture is the scientific management of lac insects and their hosts as well as pest management, cultivation, and harvesting of lac. Economically, lac of culture is a stronger option for self-employment, and many villagers, including tribals, are in this business. Lac production in India is the highest in the world. 17.11 Term